Welcome to AP Biology. Today I want to talk to you about fermentation and anaerobic cellular respiration. These are both processes that don't use oxygen. So anaerobic cellular respiration has an electron transport chain, just like we have with aerobic cellular respiration. The difference is that it has a different electron acceptor. So as the electrons move from carrier to carrier to carrier, they still are going to pump protons. They're going to use the energy from the electrons going to more and more electronegative molecules or atoms, and they're going to use that energy to make a proton gradient, just like we saw before. And that proton gradient is potential energy that can be used to make ATP synthase work, right? So it can be used to make ATP. The difference is that the final electron acceptor is different. So in this particular electron transport chain, the final electron acceptor is nitrate, but in another it might be sulfate. It might be some other atom that can pull electrons off. One of the other differences you'll see is that because this electron acceptor isn't as electronegative, right? Oxygen is the second most electronegative atom at all, anywhere. So it's actually able to have a longer electron transport chain. So this electron transport chain is going to end up being a little bit shorter, which means you're not going to pump as many protons, but you'll still make a proton gradient. So it's a little bit less efficient. But if you live in an environment without oxygen, then this is a really fantastic adaptation. All right, another process that you can do without oxygen is fermentation. And we actually do this to some degree. So there are two types of, in some cells, there are two different types of fermentation. Alcoholic fermentation, which humans do not do, and lactic acid fermentation, which we do in our skeletal muscle cells and I think in our red blood cells too. So this is a process that you can do when you don't have enough oxygen. So here's glucose, and during glycolysis, you end up making pyruvate. That's the end step, right? And you end up making 2 ATP altogether, and you take the electrons from glucose and put them onto NAD to make NADH. So glycolysis is this process, and it makes a little bit of ATP. Fermentation doesn't make any ATP at all. So why bother doing it? Well, if you do fermentation, you can stick the electrons right back on the molecule that you made. So this NADH can stick its electrons back on pyruvate, which converts it to this molecule called lactate. And when that happens, you remake the NAD+. If you remake NAD+, you can go ahead and do more glycolysis. But let's say we didn't do any fermentation. Let's say we weren't doing any of this stuff at all, right? What you would end up doing is the glucose would be broken down to pyruvate, and you would keep doing that until you ran out of NAD. If you do this process enough, you're going to have no NAD left. If you have no NAD left, you can't keep doing glycolysis, right? So you would run out of NAD, and then you'd be done. You couldn't make any more ATP. So the whole point of fermentation is to be able to remake NAD+. That's the only reason that we do it. So some organisms can do this type of fermentation and some can't. It's just another metabolic pathway. There's another type of fermentation called alcoholic fermentation, which is what yeast can do and some bacteria. And so yeast are kind of cool because they can switch, they're only one cell, right? So they can switch their form of making ATP, their metabolic pathways, from aerobic cellular respiration to fermentation. So this part is glycolysis. You take glucose and you put some electrons on NAD+ to make NADH. You make some ATP, and then your waste product is pyruvate. Alcoholic fermentation is a little bit different than lactic acid fermentation in that you rip off 1 carbon dioxide per pyruvate. So you go from a 3-carbon molecule here to a 2-carbon molecule here. You make acetaldehyde, which then gets converted to ethanol when you take your electrons off of NADH and remake NAD+. And so this part is fermentation. Right here. So again, the whole point of fermentation, the only point is to make any more NAD+, so then you can go ahead and keep doing more glycolysis. All right, a couple of vocab words here: obligate anaerobe versus facultative anaerobe. An obligate anaerobe is an organism that carries out something anaerobic, either fermentation or a type of anaerobic cellular respiration, and it can't survive in the presence of oxygen. Facultative anaerobe can go either way. It can either do aerobic cellular respiration or fermentation, some type of anaerobic process.